Homeobox genes were first discovered in Drosophila, more commonly known as fruit flies, when scientists found genetic mutations that resulted in the transformation of one body segment to another. In particular, after mutations in the antenna pedia homeobox gene, the fruit fly grew second legs in place of the antenna. What's interesting is that developmental genes don't actually code for specific sections in the body. Rather, they activate or inhibit genes that code for these sections. So think of them like the mastermind behind the operation, or a blueprint for the body. Pulling the strings, yes, but doing the grunt work? Not really. Because of this, these developmental genes work in a sort of hierarchy, as the activation or suppression of one gene may result in the activation or suppression of another, which may result in the activation or suppression of another gene, and so on and so forth, working like a domino effect. We call this sequence a gene cascade. Interesting fact, by default, most developmental genes are actually suppressed. So, to activate the gene, you first need to inhibit the inhibitor, a double negative, and to deactivate the gene again, you need to inhibit the inhibitor of the inhibitor, a triple negative. So how do homeobox genes upregulate or downregulate the expression of other genes? Well, when expressed, these homeobox genes code for transcription factors, which are proteins that control the rate of transcription of the target gene. Transcription basically involves copying sections of a gene's DNA sequence into messenger RNA. In short, the DNA nucleotide bases are copied by an RNA polymerase, an enzyme, into the corresponding RNA bases. The DNA is copied into mRNA to prevent damage to the cell's genotype and potentially causing unwanted mutations. Think of the DNA like it is your birth certificate. If you wanted to show it to someone, you wouldn't want the original to be lost or damaged, so you would photocopy it. In this case, the copy of the certificate would be the mRNA, and the photocopier would be the RNA polymerase. So now that we have a copy of the DNA, what we effectively have is the instructions to create a protein. This is where translation comes in. Here, the newly copied RNA strand is decoded, and based on the genetic sequence, amino acids are joined to form a protein. Think of it like the RNA strand is being translated into a protein. These transcription factors can either be activators, where they upregulate or increase the expression of the target gene, or they can be repressors, where they downregulate or decrease the expression of the target gene. Activators work by binding to the target gene and helping RNA polymerase to bind to the DNA strand to begin transcription. On the other hand, repressors work by binding to the target gene to block RNA polymerase from binding and starting transcription. So, if these transcription factors increase the rate of transcription of a gene, then there will be more mRNA strands, or copies of the gene available to be translated into proteins. As such, the target gene will be expressed more often, or upregulated. Of course, this can work both ways. If these transcription factors decrease the rate of transcription of the gene, then there will be less mRNA strands to be translated into proteins, and thus the gene will be downregulated. Either way, the organism's phenotype will change. A transcription factor common within most invertebrates, vertebrates, fungi, and plants is the homeodomain. A protein scientists discovered was coded for by approximately 180 base pairs within most homeobox genes, which would bind to specific DNA sequences and affect the expression of that gene. So how do homeobox genes cause genetic change? Now that we know how homeobox genes work, now we need to know why these genes can result in genetic change. While homeobox genes aren't directly involved, or don't act as the catalyst for genetic change, because of their nature as the morphological blueprint for the body, and its prolific use of gene cascades, where the change in one gene will affect the next like a domino chain. Even a small mutation in one homeobox gene can result in massive changes in the organism's genotype, and consequently, their phenotype. Think about it. If there is a mutation in one of the initial or primary gene cascades, there is a high chance of this altering the expression of all the other genes in the cascade. If we compare a single point mutation in a normal gene to one in a homeobox gene, we can see some pretty drastic differences. Studies that have been concluded that have altered or substituted homeobox genes within Drosophila and knockout mice, named so because portions of the genome were knocked out by substitute DNA sequences, in most cases found large changes in their phenotype, to the point where in one study involving knockout mice, almost 15% of the mice with their genomes altered were unviable, or unable to develop as an embryo. In the study itself, they were aptly named lethal mutations. Even if a single point mutation were to result in a missense or even nonsense mutation, usually the other cells can compensate for the cell's lack of function or dysfunction. As homeobox genes are usually expressed prenatally, Mutations often result in congenital malformations of limbs, organs, and facial features. For example, synpolydactyly, or the fusion of digits, due to a mutation in the HOX-D13 gene, and the hand-foot genital syndrome, due to mutations in the HOX-A13 gene. Most individuals affected experience abnormally short thumbs and big toes, short feet, and the fusion of bones in the wrists and ankles. While some studies have found that most homeotic diseases render the organism sterile, in rare cases, though unlikely, some mice have lived to become viable adults capable of passing on the genotype to their offspring. Since mutations in the Homeobox genes can be passed on, just like any other gene, how can this affect evolutionary change in an organism? Well, to figure this out, we need to take a step back. Historically, scientists have debated between two theories of evolution, gradualism and punctuated equilibrium. Gradualism states that the evolution of a species, or speciation, occurs primarily from the accumulation of small, gradual changes in the species. This lies in contrast to punctuated equilibrium, which states that species will undergo minimal evolutionary change for most of their history, only undergoing rapid bouts of speciation due to selective pressures caused by rare geological events. The primary cause of the debate between the two theories was the fossil record. Gradualism stated that evolution and speciation of a population was a slow and gradual process due to the slow accumulation of mutations in the genome. However, the fossil record not only showed rapid changes in the species, too fast for these mutations to accumulate over the time span that scientists estimate, but there were also gaps between species within the fossil record. According to the theory of gradualism, the fossil record should show intermediate species that possess features of both populations. For example, in the case of dinosaurs with teeth evolving into birds with beaks, an intermediate species would be present in the fossil record, perhaps possessing both beaks and teeth. Recently, however, due to the discovery of Homeobox genes, scientists have been increasingly able to address this contradiction. As discussed previously, while a small mutation in a normal gene may only result in small changes in the organism's phenotype, even a small mutation in a Homeobox gene can result in large changes in the morphology of the organism. Taking it back to dinosaurs and birds, if there were a mutation in a gene that organized facial structures that may have potentially decreased enamel production in favor of keratin production, for instance, that would have caused a sudden shift from teeth to beaks. This would have occurred in a span of one or more generations of offspring, which is a very rapid change, thus supporting the theory of punctuated equilibrium. So, to answer the overarching question, what is the role of developmental genes in evolution? Well, developmental genes enable sudden and rapid changes in an organism's phenotype, as even a single mutation can affect the entire gene cascade. This plays a beneficial role in the evolution of species, as genetic diversity in the species increases, so that's the chance that when a selective pressure is applied, there are individuals with favorable traits that are able to survive, thrive, and produce offspring and pass on those traits, furthering the survival of the species.